Hey folks. My name is Abhi. I do ML research at the ARC Institute. It's a bio and ML research institute. Specifically, I work on building models, deep learning models of cellular state. Can you look at a cell and predict how it might react in certain situations, say under application of a drug or CRISPR gene knockouts, or different ways you can stimulate the cell? Today I'm going to share a little bit of work. Actually, mainly it's just issues I faced scaling state to massive data sets that are coming out today. I have some co-authors in the crowd, so I'll shout you guys out. I want to start by calling out AlphaFold, actually. AlphaFold was the first example of seeing a machine learning model that could reliably generalize outside of the training data to any capacity. And this came out in the first year of my PhD, so it was a pretty defining thing and inspired me to work on ML for bio. And what it shows is that you can map protein sequence to structure. This is an instance level problem, but it enables scientists to assess the effects of these mutations in protein sequence and then try to simulate what would the structure be as a result. Or in certain cases now with AlphaFold 3 and other frontier models, simulate biomolecular interactions. And so it enables the scientists to perform counterfactual predictions. That's really important for really iterating fast in science. And what about the equivalent in cell models? An in silico cell that could reliably predict how cell state would change as a result of a drug would accelerate therapeutics development. For example, you could take a cell set or a population of cells that's diseased and try and see if you can induce it to look more phenotypically young or healthy. Or you could take old cells and try to make them look young, right? And there's a lot of difficulties in doing this actually with the eukaryotic cell. And so you'll see a buzzword of virtual cell thrown around here and there. And this has been done before already using more dynamical systems approaches and modeling the physical different properties and processes that go on in the cell, specifically in prokaryotic cells and more simple cellular organisms. Doing this for human cells is obviously the holy grail, but it's difficult to scale these dynamical systems to the complexity of the eukaryotic cell. And so at ARC, we're looking at how can you use deep learning to try to simulate some of the effects of the eukaryotic cells. And there's companies today investing a lot of capital into this problem. So New Limit, for example, is trying to predict what genes can you upregulate or activate that can be expressed to then help reverse aging. Xaira Therapeutics is now launching its virtual cell initiative. And by the way, this is just these are random companies. It's but Xaira Therapeutics is trying to integrate virtual cell programs as well. They're trying to integrate them with their protein models actually, but they're building out kind of the single cell side of things. Tahoe Therapeutics is another recent startup that just raised their Series A, but they're focused on repurposing drugs on cancer biopsies and training virtual cell models there to see if you can predict the effects of drugs on at a sample-specific level. And you see this in AlphaFold in different protein models. You see a lot of different people converging to similar architectures, and there's a sort of commoditization of the machine learning there. Cellular perturbation models have not had that sort of foundation model moment yet. There's a lot of reasons as to why, and some of the main ones are there's lots of technical artifacts in the data. AlphaFold had the benefit of having painstakingly and expert curated protein structure data deposited into the PDB with really, really high quality control. Single cell data doesn't have as much quality control, and a lot of times metadata is inaccurate. There's what we call batch effects, where different reagents can be used for different experiments. You know, the way you measure the cell, honestly, the capture chemistry itself can change based off of what technology you use. These introduce different biases into the data that the machine learning practitioner receives. And these limit the ability to train a foundation model on all available single cell data and then use that for transfer learning in downstream applications. And so the analogy is similar to in NLP, when before people trained BERT on all of internet text, people typically did bespoke analyses with small models trained for their specific tasks. And so when trying to predict how a cell would change, we think of it as you can break it down into 3 different parts. The first is if I add a drug to a cell, there's going to be some true effect of that drug. Maybe it kills the cell, or maybe it restores some function of the cell. there's going to be some heterogeneity or variance based off of the cell state itself, right? Different cells can be in different parts of the cell cycle. A cell could be in mitosis versus, I don't know, some other part of the cell cycle. And then there's technical noise, different capture chemistries, different readouts. And so in our paper, we focus on what we think is a tractable task of maybe one step towards virtual cells, and that's focusing on gene expression. So when you measure out a cell at cell resolution, what you're actually looking at is sequencing that are then aligned to a genome. This is all upstream of what I do. Actually, Noam does a lot of this stuff at the ARC for us. But you align these to the reference genome, and you get these histogram featureizations of cells. This basically tells you what genes are more expressed versus what genes are less expressed. And so as you think about these cells, you can think about them as histograms, and the distributions of what genes are expressed are changing over time. So when you say you want to model a perturbation, what you actually are doing in our case is predicting how these gene expression distributions change for each cell. So a lot of the data that's been collected so far for this, I would say it's in the low data regime. You can look at this plot here that shows over years the mean, or maybe it's the median number of cells per publication. And this is increasing at an exponential rate. So Tahoe Therapeutics recently put out a data set that has 100 million perturbation data points. Just recently, Parse actually put out a 10 million single cell data set, and Xaira put out a 10 million single cell data set too. But if you talk to some of the PIs at ARC, they got into the field when measurements were of like 200 cells or like 500 cells, right? So you're seeing this exponential increase in the amount of data available. And this has led to inconvenient and unscalable tooling. There's been a great standardization in the single cell field, tools like ScanPy that really simplify analyses for someone like me coming into the field who doesn't know much single cell. But the fundamental issue is that when your data is so small, it lets you fall into bad practices because it's easy. And so in the case of single cell, a lot of the tooling behind it assumes you can load the entire data set into memory, for example, or you can subsample different parts of that data set really quickly because it's in memory, right? So these are what I would call unscalable, examples of unscalable tooling. The issue is that there's, I think James pointed out, a sort of a data flywheel, but we're seeing this in our field as well here, where advances in AI, biology, and also capital investment are signaling what's going to be a really painful inflection point for practitioners in the field. So you have first biologists that are working on these breakthrough assay developments, right? Tahoe is able to do 100 million cells in a single experiment. That's kind of unheard of. And those training data are then used by people to train better machine learning models. It both enables the design of better models, and then also you're just putting more flops and data into the problem, right? So you get increased model capabilities. And once you have this better AI for biology or these increased model capabilities, that leads to more capital investment. A lot of that is due to hype, but also just like the capabilities of the models are improving. And this capital investment is, so here's some examples, right? Tahoe puts out the 100 million data set. That enables us to train models and see the first signs of what we think are interesting scaling laws in single cell. And then you see companies raising funds and or companies raising Series A and different VC companies raising funds to put back into the entire process. What that means is that, oh yeah, there's a lot of these companies now. And so what that means for the future, right? The previous generations of these data sets were thousands of cells to maybe hundreds of thousands of cells. And now the current generations of data sets are in the millions of cells to hundreds of millions of cells. And in our experience, we've seen this as kind of like a pre-BERT era to like a GPT-1-ish leap in capabilities. The next generation of data sets is going to be in the billions of cells and several billions of cells. To Noam's point, I think people are sequencing trillions of reads now. And to enable that GPT-1 to GPT-3 level leap, we need better tooling that can accommodate for these billions of cells. And so for the rest of the talk here, I'll talk about what was the pain points in going from these data sets that were meant, or these methods that were meant to handle hundreds of thousands of cells to going to hundreds of millions of cells. So state is the machine learning model that we worked on for predicting perturbations. It's trained on 3.6 million tokens, 3.6 trillion tokens, which equates to around 10 terabytes of tabular training data. For those in the single cell field, it's not all perturbation data. So a lot of this is learning the characteristics of single cell gene expression patterns in what we call like a pre-training phase. And then you train it on perturbational data after that. And the key innovation in the state paper was, can you group together similar cells? When I say similar, it means they're matched on some covariate or some metadata, and pass that into a big transformer model to then predict the treatment or perturbation effect. There's a couple reasons why we did it like this. We found that homogenizing what goes into the forward pass of the model over here helps the model learn conditional perturbation effects better. So if the model is always exposed to, say, heart tissue cells as the input, then it's able to learn perturbation effects better than if the input batch had heart cells and kidney cells and spleen cells. We find that really messes up the training dynamics and makes it more difficult for the model to learn. So already I've made some assumptions about what operations I'm able to do with my data here, right? The first issue is when you have 10 terabytes of tabular data, you can't load that all into memory, and you can't do these group by operations on specific metadata. So we switched from AND data to using H5Pi. This is basically just memory mapped data of the different gene expression histograms for these cells. Getting a little bit into the weeds of the model, but when you're working with single cell data, the measurement of the cell is actually destructive to the cell itself. So you're never measuring the same cell pre and post perturbation. So instead, the correct abstraction that we think to make is comparing distributions of cells. So you have a bunch of cells that we call control cells, and you measure their gene expression profiles. And then you have a separate set of cells that receive some drug treatment. And you then try to train a machine learning model that can translate one distribution to another distribution. This is why this idea of metadata matching becomes really interesting, because you want to learn a conditional transformation across these distributions. And so for each one of these forward passes and each get item call of the model, where you're picking a specific pair of cells to learn this effect on, you have to actually sample a metadata matched cell, like the same cell type or from the same donor, for example. So this turns out to be really slow. And you can hack away at it a little bit, which is what we did at the state paper, right? So given a heart cell, you can cache in your data set what are all the indices within this data set of all the other heart cells. And then you can randomly sample from one of the other heart cells. And it works when you're segmenting indices for very simple group by conditions, right? If you want to stratify by cell type, or if you want to stratify by donor. Where this issue starts to become a little bit more of a problem is in the future generation of models, where you want to stratify by more complicated conditions. So if you want to stratify by tissue and by donor, or by cell type and by donor, you're having to store a lot of things into cache and having to segment the indices a lot more. In our case, because in state we focused mainly on learning cell type specific effects, we only had to stratify the data by cell type. And we made a repository cell load that basically all it does is it takes AND data and converts all of the strings into integers, like unique integer representations. If you use H5Pi, these are just the codes in H5AD files. And then caches the metadata. And we found that this led to a 10,000 times speed up in runtime on the naive approach translating from memory mapped AND data here. So it works in the case of the 100 million cells and actually reaches within 2x the runtime of just having the entire AND data in memory. So the overhead of moving to this approach is not that bad in favor of scaling to larger data sets, even when in-memory approaches are feasible. Evaluation is a completely separate issue from training, though. So training in deep learning, you can do things mostly at the batch level. So all you need to do is load in one batch from disk into memory, and then you do your model, you do your backprop, and you're good to go to the next batch. But evaluation in single cell is a whole other beast. So a lot of traditional single cell evaluation methods require loading the entire data set into memory. sometimes this is really not needed because a lot of the metrics that are actually being computed actually just take population averages. And so you actually only really need the average sometimes. You don't need to load every single point in your tabular data. There were a couple tricks along these methods that we did. One big one was also differential expression analysis. So some of these statistical tests can be parallelized across multiple threads. And so we implemented some of these in our paper to scale evaluations to, I think, 20 million cells for the Tahoe data set. So cell eval is a package actually that Noam wrote, and it took our eval time from 72 hours down to just 30 minutes. And so some takeaways from this, whenever possible in your single cell workflows, use GPU acceleration. NVIDIA is investing a lot of money into this and a lot of engineer resources. So it was helpful for me to use this whenever sanity checking or doing such. Use half precision whenever possible for your evals. A lot of times you don't need full precision and it cuts your memory by a lot, especially as you work with evaluating on some of these data sets. But also be careful when casting to NumPy because half precision in NumPy is really jank and not as well fleshed out as in PyTorch. And then lastly, parallelize your operations whenever possible. It's really difficult to fit everything into memory, but if you can split your evaluations by cell type or split your evaluations by condition, then you can just parallelize that across a lot of workers. And a lot of times I find that single cell people are a little hesitant to do this, but in our case, taking some of these issues and dealing with them at the beginning of our workflows enabled us to move really fast in the development of state. So the Tahoe 100 million single cell data set was provided to several frontier research groups at the same time. And our approach to hacking on this data set was to spend a miserable 3 weeks on just setting up the tooling and how do you parallelize things as much as possible? How do you write scalable data loaders that are memory mapped? How do you do this metadata caching? But it really unlocked rapid progress. Anyone that's building deep learning systems knows that if your evals take 72 hours versus if they take 30 minutes, it's just a different league of how many ideas and model architectures you can hack around and try. And so if you're building these deep learning systems, build that stuff out first, and then optimize for your ability to feed data into the model. In our case, we found that following those two guiding principles of evaling fast and optimizing for data throughput in the model led to state-of-the-art performance across a couple of different metrics on different data sets. I don't want to go too much into the metrics here, but this one here is Pearson correlation. This is basically just what is the Pearson correlation of the gene expression you predict versus the observed data. You see some interesting scaling here as well. So on this right side here, we have the smallest data set, which is in the hundreds of thousands of cells range. And you see that state, which is this blue bar here, does similar to other methods, including other simple linear baseline methods. Once you move to the signaling parse PBMC data set, so this is signaling cytokines that are added to the cell, you see a larger improvement. So here the data set is 10 million cells, and you see a 47% improvement on this metric as compared to other baselines. And finally, on the Tahoe data set, you see, in this case, a 63% improvement as compared to other baselines here. So what's interesting is that building the tooling that enables these models, it's an interesting relationship between the two of them. First of all, you need more data to even differentiate what is a better machine learning algorithm. Like, we would not have known that state is a better algorithm on some metrics if we were just looking at the smaller data set. But in order to do that, in order to look and analyze at these larger data sets, you have to put in the investment into the painful data loaders and all the unglamorous start. And then this is just another metric here. This is a ranking of effect size of perturbations. This is basically if you apply a perturbation to a cell, how many genes are significantly up or down regulated in their expression. And this is like basically a ranking correlation statistic on that. And again, you see this sort of scaling as you increase the size of the data set. We see that state is better able to leverage the data. So looking forward, several different institutes have pledged to reach 1 billion cells. ARC has its own virtual cell atlas that it's growing right now, both in terms of in-house data and also an AI agent that we wrote that automatically curates data from publicly available repositories. Chan Zuckerberg has also launched its own 1 billion cell initiative. And then you have the private players, right? These guys are raising a lot of capital to do data generation as well. And so several billions of cells with perturbations are going to be available. And there's a lot of shortcomings in the methods that we wrote in developing state. And so one issue in, I think, some of these communities is there's no commoditization of this tooling. For example, when we were writing state, we wrote all of this stuff from scratch. But there have been similar efforts and other really good libraries for building some of these single cell data pipelines. So that's the first thing is there needs to be some commoditization and collective effort on building good tooling that everyone can use so people don't have to reinvent the wheel. In terms of cell load, there's a lot of room for improvement. So H5Py natively right now doesn't support concurrency. And this leads to a big issue when you're training on a lot of different files. You can look at this really horrific GPU utilization for state. You need something that allows concurrent access. And so this is something we're actually actively exploring at ARC now, is like, what is a system that enables better GPU throughput as you scale more data? And you also are going to need more complicated group by operations, right? A lot of what people are looking at now is like, how do you mix in natural language with some of these perturbation models? That sort of thing requires knowing the metadata about the cell, harmonizing the metadata about the cell. People are using AI agents to do some of this now, but the key takeaway is the systems itself, you can't cache every single possible group by. So you have to actually build better systems to load data on the fly. And lastly, I put a lot of blame on the tooling and the data, but there's this quote I really like. So AlphaFold was trained on largely the same data as AlphaFold 2. And Nobel laureate John Jumper says that AlphaFold 2 was mainly just better machine learning. And that led to winning the CASP competition and significant improvement over AlphaFold 1. So while there is a need for better tooling and better data, there's also a dire need in single cell for better machine learning methods as well to really make use of this next generation data. And then I just want to thank my collaborators. So this was a huge effort. Yusuf, our group lead, who's not here, but Dhruv, who's here, Noam. I think I saw Lishba earlier as well. So thanks to all my co-authors, and thanks for having me, Shreya and Kenny.
Probably 2 questions, because it's getting late. Everyone wants to eat pizza and stuff.
Hi. How do you see these models being used? Is it just a massive search that then tells you where to look, or do you see these models actually providing mechanisms and pathways?
I'm sorry, I can't hear.
Hello? Hi. Yeah, so I just wanted to ask, how do you see these models being used? Is it just a massive search that then tells you, where to look specifically? Or do you see these models actually providing you mechanisms and pathways of what kind of effects these perturbations are having?
Yeah, so the question was, how are these models actually going to be used? And is it to search across maybe the space of perturbations, or is it to uncover underlying mechanisms of biology? I think the first application is more so search. I see a more direct path to search than understanding underlying biological mechanisms. Maybe the next generation of data will unlock some of those capabilities, but search is a problem I think is more tractable now. So like, what is the perturbation required to induce a specific phenotype? This is what companies are doing right now, right? So I think that's the primary use case I see.
Hi, Abhi. Thanks for the talk. I was actually interested in your thoughts on some of the benchmarking results that you showed, and also what was showed in the paper. So obviously, state was outperforming the other baselines in most of the cases. But I was interested in the signals that you got for the benchmarking that's based on the differentially expressed genes, which are more often the things that we are actually interested about, because they are the genes that respond the most strongly to the perturbation, as compared to the transcriptome-wide ranking of the signals. And I think, if I remember correctly from the paper, the absolute value of the benchmarking results was better for the transcriptome-wide ranking as opposed to the DEGs themselves. So do you think that the strategy of training on cell sets might actually kind of reduce the the ability to actually predict things that we care about? You get better predictions at a genome-wide level at the expense of the extreme levels that we actually care about, and also what's the implication of how we think about how we should work with data quality and things like that in that sense, and machine learning model design.
That's a good question. I don't think you would want to sacrifice on genome-wide resolution. I think the goal is to build a model that is capable of doing that. I think what you get from the cell sets is a denoising, essentially. By being able to look at more cells at once before you make a prediction, the model can probably drop out bad cells or something similar to that. And I think that the reason that the ranking might be down is maybe actually not so much due to the cell sets, because we saw that that metric was not as variant when we varied cell set. I think that might actually be a separate issue from the distributional training on cell sets. I think the Spearman ranking is something we want to look into more, though. I agree it's probably the metric that a biologist would look at the most, right? Spearman, you're talking about the log-fold change Spearman rank?
Yeah.
Yeah. So, yeah, it's a good question.
Thank you.
Yeah.
Yeah, I was curious about the wall clock time for your training run, and also, how many nodes, how many GPUs, that kind of stuff.
Oh yeah, sure.
Share some
So for the smallest data sets, you can train a model to convergence in, oh wow, they're really partying. You can train a model to convergence on the genetic data sets in I want to say 3-4 hours. And that typically entails just on one H1. It's really low utilization actually, because the model size itself is not very large. Most of the time comes from just, and on the signaling perturbation data set, that was in the next scale of maybe 10 million cells. That took around a day, usually. So not significantly longer. And Tahoe Tahoe was heavily I/O bottlenecked, so you'll see that in the GPU util, right? Even that model doesn't require so much VRAM, and we were usually able to train each Tahoe model in about 2 days. Yeah, and so that would be for, it's actually, the model really struggles to overfit a lot of the time, which is good. I think there's a lot of inductive bias we're baking in that prevent that. On Tahoe, it was usually around 400,000 optimization steps. Yeah. So every single perturbation run here was single GPU. You only need one, and you can probably train it on like an L40. Definitely, yeah. Yeah. Okay, thanks.